This is Jose Merino, Editor-in-Chief of the Neurology family of journals. The Neurology podcast provides practical information to neurologists and other clinicians to help them provide better care for their patients. Thanks for listening and have a great week.
Hello, everybody. My name's Andrea Gropman. I'm the Director of the Neurometabolic Translational Research Initiative at St. Jude Children's Research Hospital. And today I have the pleasure of interviewing Stefan Pulst. Stefan is the Chair of the Department of Neurology and Professor of Neurology and Human Genetics at the University of Utah. Stefan is the founding editor emeritus of Neurology Genetics. Welcome to the podcast.
Thanks for having me.
So we're all very interested to hear about the role that neurology genetics has played in advancing our understanding of rare neurologic diseases and their genetic underpinnings.
Yeah, let me take you back to 2014 when Bob Gross, the then editor of Neurology, approached me to think about a new spoke journal, a subspecialty journal, following the steps of Neurology N2. And I was very enthusiastic and wrote him back, and he thought that I would make a good editor-in-chief for this new spoke journal. And so we published our first article actually in 2015. And it has filled a niche for clinicians and geneticists really to bring genetic variants and their phenotypes together, and has done quite well. We are now listed in all the major indexes in PubMed. We have a very good impact factor for a subspecialty journal. And it has been a pleasure to guide the journal through the first 10 years of its existence.
You recently published an editorial on the event of the 10th year anniversary, and it touches both on your reflections and inspiration for the future. What inspired the structure and themes of that 10th anniversary edition?
We were approached by the publisher and thinking that would be a nice way to end the first 10 years. And in the end, it was really a personal selection of 6 reviews that I thought balanced personal reflections, gene discoveries, application of testing of common diseases and gene-based therapies. And this anniversary issue starts out with 2 personal stories by one of my associate editors and personal friend, Massimo Pandolfo, about the discovery by his group in 1996 of the Friedreich's ataxia gene, and personal reflections on my part of the discovery of the SC2 gene in 1996. And we both follow the long journey from gene discovery through modeling of disease to now finally finding some of the first therapies that may modify the course of the disease. The other 4 reviews in that anniversary issue cover genetics of common diseases like Alzheimer's disease and epilepsy, both from the Mendelian side and the risk allele side. One review talks about gene therapy, and finally a overview of the UK Biobank, a population-based approach to population genetics.
And in the editorial, you also talk about the progress that has been made in understanding the genetic architecture of various neurologic diseases. What aspects of this progress do you find the most promising, and why?
Really, the last 10 years have seen enormous progress on multiple avenues. One is sequencing technology. We have gone from expensive Sanger sequencing base by base to whole exome sequencing to whole genome sequencing. And really lately, the great revolution has been long read sequencing, long read whole genome sequencing. And that has resulted in finding novel repeat diseases. Just in my particular area of expertise, the ataxias canvas was identified, probably now the most common recessive ataxia, followed by SCA27B, an intronic repeat expansion. Again, long repeats, similar to Friedreich's ataxia. And we recently discovered SC4, a novel repeat class, probably a rare disease, but a GGC repeat that could only be identified by long-read whole genome sequencing. So that really has changed the discovery of certain repeats, probably in the future also of complex genomic rearrangements. And that has been paired with bioinformatic approaches to actually being able to detect repeats, for example, in shorter read sequences by using bioinformatic tools. So I think that both bioinformatics has changed, the sequence technology has changed. In terms of genetic testing, we now see a move from panel testing to whole exome testing, which provides so much more information. And we see whole exome now transitioning really quite seamlessly into whole genome sequencing, which provides even more information. And that is coupled actually by a lot of the testing companies with very good bioinformatics approach that is meaningful to the clinician. So the clinician can get some ideas about which variants are actually meaningful in terms of the disease. At the same time, we have seen, and that's why I chose this one review on the UK Biobank, a really very novel approach to near population-based genetics. And I say near population-based because it's not truly yet population-based, but 500,000 citizens in the UK have participated in this, accumulating large phenotypes, both clinical, molecular, imaging, combined with genetic information. And that has really changed our insights on causation of disease. But what I find very interesting, since I'm interested in repeat disorders, it has also told us that we have largely overestimated the penetrance of many diseases. When you look at a population base and not an academic center base, you find that some mutations are much less penetrant, which I think is very important. If these days an individual gets a diagnosis of a C9orf72 repeat expansion with a looming diagnosis of ALS or FTD in the future, I think it makes a world of difference to know what the age-dependent penetrance is.
And how can clinicians use that in their practice, for example?
We now realize we should sequence much more. I think compared to a repeat MRI, we all as practicing clinicians know that for many degenerative diseases, not that much changes from MRI to MRI on a yearly basis. Sequencing is now becoming less expensive than doing yet another MRI. And I think it provides an enormous amount of information, both giving the disease a name, but also allowing reproductive choices. And the great hope still is providing personalized treatments. And we are seeing this now in a variety of degenerative Mendelian diseases where ASO therapies or small molecule therapies are making a difference.
And as precision medicine becomes increasingly prevalent, as you alluded to, do you see the journal Neurology Genetics somehow contributing to its implementation in neurology practice?
Yes, very much so. Really, this has been, I think, the bulk of our articles has been description of genetic variants, phenotypes, sometimes novel variant in an established phenotype, sometimes a novel variant and a novel phenotype, which is, of course, the most difficult to discern. Are they both really related? So the evaluation of variants has become very important. And in the last years in neurology genetics, we really follow the recommendation of the American College of Medical Genetics to classify variants so we know what evidence there is. And it's really not black and white. It's really adding on evidence to arrive at the likely conclusion that a variant is pathogenic. Actually, pathogenic variant is now the new name for what was used to be called a mutation, just to make it clear that there is a gradient of impact of variants. And I think neurology and genetics has really played an important role in publishing reviews of larger case series of genetic diseases, but also doing case reports of interesting aspects, including sometimes just interesting imaging associated with genetic diseases. And very often in retrospect only do we recognize that there were actually telltale, rather pathognomonic signs available that we really never quite realized. A good example is an article that just came out in Neurology Genetics this February on a very particular sign in the MRI of the posterior fossa in ST27B. Clearly, we had all seen these patients, but we could never quite realize that it correlated with the presence of this repeat mutation.
So this journal has really filled a niche in this rapidly growing field. And in your editorial, you also emphasize inspiring the future. What excites you about the next decade in neurogenetics? Do you see any trends or areas of research that are standouts?
I'm very excited about population-based genetics because we will realize that some genetic variants are predisposing variants or risk alleles that can only be recognized if we look at large-scale population data. We will be able, and we have already done this, look at variation of normal traits, let's say hippocampal volume, not looking at dementia or mild cognitive impairment, but looking at hippocampal volume as an intermediate marker and then asking what kind of genetic predispositions determine hippocampal volume. And of course, the UK Biobank also offers the unique opportunity to look at environmental factors and their interaction with genetics, which I think will be one of the most complex, but also one of the most interesting aspects of genetics. I think the other area is the long-held promise that we have given our patients 30 years ago that we find the disease gene, we will find a therapy. We have learned that it's much harder than we thought. Even those of us who were quite experienced in developing therapies, finding a disease gene does not mean that we can easily undo its damage or replenish its function. But we have a lot of tools in our hands now that will allow us to at least try to approach these genetic diseases.
What challenges do you see in the next decade when it comes to therapeutic interventions like you mentioned?
I think we need to realize it's not magic. Genetic therapies are just like any other therapy, and we will go through trials and failures. And like small compounds, many compounds will have side effects, unanticipated side effects. We will rediscover again that mice are just mice. They're models of disease, but mice are not humans. So we will need studies of non-human primates, and we will not get around trialing our new compounds, be they biologicals or small compounds, in patients, and do that very carefully.
And just in the broader perspective, how has the community of contributors and readers shaped the direction of neurology genetics over the years? And what's that meant to you personally as an editor?
It's really a great question, because I really, as an author and as an editor, I had not looked at this as a community enterprise. And it actually really is. In our role as authors, we often see our relationship with the journal as potentially adversarial, in the kind of, they just don't like my papers, or they don't like me or my laboratory. And on the editor side, the viewpoint is we want to make sure that we publish reproducible science, and at the minimum, an exciting, novel, and relevant science at the best end of the spectrum. And what connects both of these communities are really the reviewers. And I found that that is really the crux of a lot of journals these days. Is it's very hard to get people to review. We are all very busy, especially very good people are very, very busy. And so one plea I want to make in this podcast is actually to engage the next generation in the review process. Fellows, clinical fellows, postdoctoral fellows, they can all work with more senior reviewers to review articles. And that's two things. It allows reviews to go forward, and it trains the next generation to do reviews in the future.
That's a very important point. As people get busier and the number of journals increase, it appears that the same people, more likely senior people, are asked to review for multiple journals at the same time. I always found as a junior faculty, I learned a lot by reading other people's articles and looking at it critically. So given that neurogenetics was a nascent field when you were acquiring the journal and building it, what advice would you give to other journals or editorial boards that are aiming to create a lasting impact in a niche but rapidly growing field?
That's a heavy question. I think one can never sacrifice editorial independence and quality. And I think I can speak for most of us. We get requests to submit articles to questionable journals. And I think it is sometimes not that easy to stand out as a novel spoke journal from this background of what is now called predatory journals. And I think the way we set ourselves apart is having a clearly visible editorial board of individuals who are experienced in their field, have uncompromising peer review, ideally also relatively rapid peer review. I'm not talking about 48 hours, but 2 to 3 weeks is reasonable. And I have always found, as editor-in-chief of Neurology Genetics, to really get involved with the authors, guide them in their responses, and read their rebuttal letters and point-by-point critiques very carefully. And I think if one follows this, one can, after a few years, have a successful journal. I think the other lesson is it may take up to 10 years to really establish oneself as a journal that has a reputation.
This has been a real pleasure. So thanks so much for your time.
This is Stacey Clardy, your podcast editor. If you've enjoyed the podcast, please take a few moments to subscribe, rate, and review the Neurology Podcast through Apple Podcasts, Google Podcasts, Spotify, or wherever you listen. And remember, you can always head to neurology.org/podcast for our full list of past episodes. Or you can also search by keyword in your podcast app for any neurology-specific topics you want to learn about.